I am Lena Kenyon, a research scientist working at Morpholino Therapeutics, LLC. My work is focused on curing human cancers. I am here to provide you with a verbal introduction to our LK1 Cancer Cure Program. It will be accompanied by a more extensive written text. LK1, an advanced versatile drug for curing human cancers, by James Semerton, PhD, Lena Kenyon, BS, Donald Moss, PhD, employed at our new company, Morpholino Therapeutics, LLC, located at 1001 Semerton Way, Philomath, Oregon, USA. Email jim@morpholinotherapeutics.com. For curing human cancers, we have developed a multifunctional, highly versatile drug designed to specifically alter functioning of a variant of the HCGB protein, commonly known as the human pregnancy hormone. We target the mutant variant of HCGB protein because of its unique properties underlying human cancers. Those special properties include: 1. Mutant HCGB proteins are present in all human cancers. 2. Mutant HCGB proteins are the central cause of all human cancers. 3. Mutant HCGB protein is essential for the viability of all human cancers. Our Cancer Cure Program. Cancers have been a bane on humanity for all of history, with devastating results. Dr. Sid Mukherjee published a popular book about human cancers, The Emperor of All Maladies. The book makes clear that cancers today are widely regarded as the worst of all diseases from which people suffer. Until about the year 1900, most cancer patients were treated surgically, but very few patients survived such treatments. Significance of mutant HCGB protein and its relation to human cancer. In the year 1920, a scientist discovered a large, very unique protein structure which was identified as playing a major role in all human reproduction, hence the name human pregnancy hormone. Additionally, a variant of this protein has also been found in various human cancers, that being mutant human chorionic gonadotropin, HCGB protein. HCGB protein is essential to the life of all humans before birth. This unique protein is involved in a vast number of functions crucial to nurturing and protecting the baby, including evading the mother's immune system, protecting the baby from disease, formation of new blood vessels, and increasing growth rate. Once the baby has developed to the point of its birth, typically 9 months, it no longer requires the HCGB protein, so fundamental to its development during gestation. Therefore, the HCGB protein is eliminated along with the mother's placenta as the baby is born, leaving only the belly button as a lasting mark. Because of that, now the mother and the child no longer have access to the HCGB protein. At this point, the baby begins to enter maturity, and as a mature human, it takes it into adulthood. Tutorial on the cause of human cancers. A. Normal HCGB protein. B. Mutant HCGB protein. The normal DNA that codes for HCGB protein in humans is activated when the sperm and the egg join together, which induces the development of human embryos and is unique to the human development only. This normal HCGB protein is only expressed during pregnancy and not expressed outside of that. In sharp contrast, in rare events only, mutant HCGB protein is expressed outside of pregnancy due to mutagenic events. Specifically, when the genes and DNA that code for the unique human HCGB protein can be mutated by a variety of sources, such as radiation, high energy UV exposure, toxic chemicals, certain viruses, etc. Once mutated, it will begin generating an HCGB variant protein. When this mutant variant propagates, it constitutes a cancer in the patient. And because of the differences between this mutation from the normal HCGB protein, typical targeting strategies aren't effective. This random mutagenic event to develop cancer is typically very rare, but with increasing decades, there is an increasing probability of this random mutagenic attack developing over time. Mutagenic events can alter the coding sequence of genes, such as the HCGB protein gene, leading to the production of faulty proteins. When these mutations occur, they disrupt normal cellular functions and interfere with the body's processes, constituting cancer. Once the coding sequence is damaged, the gene no longer functions properly, causing the affected cells to lose their normal regulatory mechanisms. The mutated protein serves no beneficial purpose for the organism and generally constitutes a detrimental disadvantage, resulting in uncontrolled cell growth or other malignant changes. If the mutation continues unchecked, it commonly has the detrimental effects of cancer and ultimately threatens the life of the patient. Structural information of normal HCGB protein. The core HCGB protein sequence is a 145 amino acid long protein. This particular HCGB protein is synthesized in a ribosome within the cytosol of the cell. A smaller HCGA protein sequence of a different structure and function is a 92 amino acid long protein. Typically, the larger HCGB protein pairs with the smaller HCGA protein to form a non-covalently linked dimeric structure. After the dimeric structures have been assembled, a maturing process begins whereby glycosyl groups, a type of sugar, are attached to a wide variety of sites along both the HCGA and the HCGB proteins. These mature glycosylated structures can enable an amazing variety of very complicated functions. Failed attempts to target mutant HCGB protein. By the 1970s, the mutant HCGB protein had been widely recognized as causing many different types of human cancers. Then, in the year 1979, a research paper was published which at that time reported that mutant HCGB protein was the key structural element which instigates human cancers. From about the year 1980 through about the year 2010, the close connection between mutant HCGB protein and a wide range of human cancer types led a great many cancer researchers to attempt to precisely target the mutant HCGB protein. This attempt led to the quite reasonable premise that targeting the cancer-causing component should block the function of the mutant HCGB protein and result in defeating the patient's cancer. Generally, such molecular targeting was attempted by using the newly developed and highly promising precision-targeted monoclonal antibodies. Typically, a human monoclonal antibody targeted against a specific protein site in the mutant HCGB protein structure. Alternatively, precision-designed small molecule drugs were also used to try and block mutant HCGB protein in cancers, but often at greater cost and complexity. The hope was to achieve a significant therapeutic effect on multiple cancer types commonly found in association with cancers expressing mutant HCGB protein. Notably, when new, more sensitive detection methods were employed, it was reported that human cancers are found to be expressing mutant HCGB protein at quite low levels at some stages and detectable at even trace levels in the cancer's life cycle. In spite of considerable efforts and large expenditure over the course of 3 decades, researchers have been unsuccessful in blocking the mutant HCGB protein in human cancers. These attempts did not defeat even a single type of cancer due to mutant HCGB protein, which include unique sugar structures with extensive glycosylation and hyperglycosylation levels, along with an extensive length of the dual dimeric proteins. The sugars from the glycosylation process shields the dimerized mutant HCG protein from attack by attempted therapeutic treatments, such as by monoclonal antibodies or small molecule drugs. The limitations of the past use of these targeting strategies is that they target the mutant HCGB protein after it exits the cell, and only after it has been glycosylated, which obscures the targeted site. These attempted therapeutic efforts are generally far too little and far too late to be of significant therapeutic value, which ultimately led to abandonment as a therapeutic strategy. These therapeutic strategies consistently failed all attempts to defeat human cancers by failing to block mutant HCGB protein. Even monoclonal antibodies, which were revolutionary at the time, were unable to overcome the targeting challenges. This led the researchers to conclude that mutant HCGB protein is far too complicated to target and therefore undruggable with the capabilities of the then current technology. That undruggable label led to a near universal skepticism throughout virtually all of the cancer research community in regard to the feasibility of targeting the mutant HCGB protein. Consequently, targeting mutant HCGB protein has almost entirely been abandoned by those in the cancer research community. However, this abandonment of mutant HCGB protein as a key target for cancer treatments was a dire misstep that has led to the stalling in the development of more effective treatments for cancer. This is because the HCGB protein is the most universal vulnerability in human cancers, and without a plan to deal with the mutated variants, eventual remission is all but certain. We contend that any treatment intended to be effective, broadly applicable, and long-lasting must tackle the mutant HCGB protein in some fashion. Following the abandonment of earlier approaches, multiple other researchers explored alternative targets and pathways to develop new anti-cancer therapies. As a result, several FDA-approved conventional treatments are now available for cancer care. Conventional cancer treatments. Currently, there are multiple other therapeutic strategies being pursued to kill cancer, some with more success than others. Targeted therapy. Targeted therapy is a type of cancer treatment that targets proteins that control how cancer cells grow, divide, and spread. It was the foundation of precision medicine. As researchers learned more about the DNA changes and proteins that drive cancer, they were better able to design treatments that target these proteins. Most targeted therapies are either small molecule drugs or monoclonal antibodies. Small molecule drugs are small enough to enter the cells easily, so they are used for targets that are inside cells. These drugs are used routinely, though monoclonal antibodies have become the dominant choice. Monoclonal antibodies are proteins produced in the lab. These proteins are designed to attach to specific targets found on cancer cells. Some monoclonal antibodies mark cancer cells so that they will better be seen and destroyed by the immune system. Other monoclonal antibodies directly stop cancer cells from growing or cause them to self-destruct. Still, others carry toxins to cancer cells. Targeted therapy does have some drawbacks. Cancer cells can become resistant to targeted therapy. Resistance can happen when the target itself changes and the targeted therapy is not able to interact with it. Or it can happen when cancer cells find new ways to grow that do not depend on the target. Because of resistance, targeted therapy may work best when used with more than one type of targeted therapy, or with other cancer treatments such as chemotherapy and radiation. When targeted therapy was first developed, scientists thought that it would be less toxic than chemotherapy, but they have learned that targeted therapy can also cause serious side effects. Hormone therapy. Hormone therapy is a cancer treatment that slows and stops the growth of cancer that uses hormones to grow. Because hormone therapy blocks the body's ability to produce hormones or interferes with how hormones behave, it can cause unwanted side effects. Immunotherapy. Immunotherapy is a type of cancer treatment that helps the immune system fight cancer, and there are several types of immunotherapies used to treat cancer. Immunotherapies can share problems found in other therapeutic approaches. Chemotherapy. Chemotherapy is a drug treatment that uses powerful chemicals to kill fast-growing cells in the patient's body. These are typically small molecule cytotoxic drugs. Many different chemotherapy drugs are available. Chemotherapy drugs can be used alone or in combination to treat a wide variety of cancers. Though chemotherapy can be an effective way to treat many types of cancer, chemotherapy treatment also carries a risk of severe side effects. Some chemotherapy side effects are mild and treatable, while others can cause serious complications. Famed cancer researcher Dr. Vincent DeVita's superb book, The Death of Cancer, describes the first major advance in chemotherapy, high-dose multi-cocktail chemotherapy. Of particular value, DeVita also describes a variety of concepts and principles that help organize the massive complexity of cancer. Also, in his book, DeVita suggests jointly targeting multiple cancer hallmarks as a promoting therapeutic strategy. DeVita and his colleagues' advances increased the overall survival rate of cancer patients an unprecedented leap from 25% to 62%. Nonetheless, these treatments can often fail to provide effective cures, particularly for aggressive metastatic cancers. The inception of innovative therapeutic technologies. The promise of mutant HCGB protein as a universal cancer target was undeniably compelling, but it was ultimately abandoned prematurely due to the technological limitations of the time. While targeting mutant HCGB protein made sense in theory, the available methods of therapeutic implementation were not advanced enough to realize its potential. Even though new high-tech monoclonal antibodies were a dramatic improvement at the time, new technology was required to overcome the failures of monoclonal antibodies. Somerton and his new collaborator, soon to be Dr. Kenyon, were unsatisfied with the current treatment options. With the determination to reevaluate the supposed impossibility of tackling mutant HCGB protein, we invited the two world's experts on the HCGB protein, Dr. Larry Cole and Dr. Stephen Butler, who jointly authored the most publications on the subject. The goal was to discuss possible alternatives to the conventional targeting strategies. Although the experts were confident about the benefits of blocking mutant HCGB protein, this meeting ultimately failed to come up with better alternatives and did not result in a suitable advance. Figure 1, labeled left to right, James Somerton, Lena Kenyon, Larry Cole, Donald Moss, and Stephen Butler. Subsequently, Somerton and Kenyon at Morpholino Therapeutics, LLC, later devised and developed a mixture of highly unconventional strategies and our newly developed technology to target the mutant HCGB protein. In sharp contrast to the conventional strategies, our ultra-precision targeted LK1 drug decisively and very specifically enables blocking of the function of the mutant HCGB protein, thereby achieving the central goal of destroying human cancers. This novel and innovative therapeutic strategy works by targeting directly inside the cytosol of the cancer cell and before the dimerization and or addition of the glycosyl groups of the mutant HCGB protein. This strategy prevents the ribosome from making the final protein structure of mutant HCGB protein, which the cancer cells rely on to propagate. Can our LK1 drug block all human cancers? Yes, our LK1 drug can cure all human cancers by leveraging our affordable, unique three-component drug structure to effectively target a crucial weakness shared by every known human cancer type. Each of the three components provides a vital mechanism missing from other anti-cancer strategies. The first component is our advanced targeting technology, which is selective to block the function of mutant HCGB protein. The second component delivers the targeting component from the site of injection into the patient, then penetrates the cell and ends its journey where the mutant HCGB protein is synthesized, inside the cytosol of the cancer cell. This allows the first component to prevent the maturation of mutant HCGB protein by blocking its function. This allows the drug to eliminate cancers regardless of the type of cancer. The third component of the LK1 drug is our unique multifunctional structural agents. These components protect our drug from degradation in the body and in the cell. These special structures even shield the drug from lysosomal degradation. These multifunctional agents also avert immune responses common to other therapeutics. Furthermore, these allow highly predictable targeting and provide several important logistical benefits, such as good aqueous solubility and stability, sufficient to persist unharmed through autoclave temperatures. Finally, it is affordable due to its efficient assembly and easy workup. Treating brain cancers. Brain cancer makes up about 25% of all cancer cases. Since LK1 is too large to pass through the blood-brain barrier on its own, we collaborated with Dr. Edward Newalt at the Oregon Health and Science University. Dr. Newalt pioneered a system which involves introducing a high osmolarity solution to temporarily premolize the blood-brain barrier, allowing LK1 to effectively treat brain tumors. This allows treatment with our technology in what would otherwise be a very difficult attempted treatment strategy. Contributors to our LK1 drug development. 1. Lena Kenyon, BS, and nearing a PhD. Lena completed her Bachelor's of Science in Chemistry in 2017 and soon thereafter joined GeneTools, where she assisted Somerton in his research. Because of her high competence, Lena was persuaded to pursue a PhD in biochemistry with Somerton's full support and while also working at GeneTools. By late 2021, she was well along in her doctoral coursework, had passed her prelims, and had settled on and carried out preliminary planning for her intended extremely challenging dissertation project titled Development of a Broad-Spectrum Cure for Human Cancers. The company Morpholino Therapeutics, LLC, then set Lena up with her own well-equipped research lab optimal for starting her dissertation project. which she began January 2nd, 2022. By taking a 1-year leave of absence from her doctoral program to spend a full 12 months highly focused exclusively on her dissertation project. By the end of 2022, she had completed, designed, and synthesized the sophisticated LK1 drug. Plus, she had obtained compelling proof of principle results showing that the drug was indeed a broad-range treatment for human cancers, that it destroys the cancers, and that it does not harm normal cells because mutant HCGB protein is not expressed in normal cells. We have named this new drug LK1 in honor of Lena Kenyon, having been the first to design, synthesize, and test a drug expected to defeat all human cancers. Additionally, after confirmation studies are completed, those very impressive research results are to be submitted for patenting and subsequently publication. Designed, synthesized, and tested the first functional LK1 drug. Continues to optimize functions and expand applications. 2. Dr. Donald Moss, PhD, carried out key studies of the HCG component that clarified the steps involved in destroying the cancers upon deletion of HCG. Provided and continues to provide a variety of technical skills, scientific insights, and a broad knowledge base in biology and particularly in neurology, which he picked up in the course of his many decades spent in inventing, developing, and testing the most effective, safest, and least expensive of all drugs for curing Alzheimer's disease. 3. Dr. James Somerton, PhD, conceived and implemented the first antisense drug strategy, November 1969. This was a new and novel breakthrough targeting genetic strategy to effectively and precisely block the coding RNA sequence, i.e., the sense RNA genetic sequence of a selected gene target. Submitted the first antisense publication in 1973, rejected by reviewer as a pipe dream. Subsequently published an expanded version of that first antisense paper, finally published in 1979 after completing proof of principle experiments demonstrating the feasibility of the antisense concept, based on his postdoc work carried out at UC Berkeley, funded by NIH. Filed his first antisense patent application in October 1978, assigned to NIH in thanks for NIH funding his postdoc fellowship at UC Berkeley. Founded first antisense company, Antivirals Inc., invented a radically redesigned antisense technology, resulting in the morpholino structural type, which dramatically improved structural properties, particularly in regard to the use for constructing novel products for special uses. 6 issued patents. For more information, see Morpholino Therapeutics Addendum 1. Founded and self-funded another antisense company, GeneTools LLC, focused on providing custom sequence antisense tools for modulating the function of any genetic sequences a researcher from anywhere around the world might wish to study. Note that GeneTools offers free access to abstracts and citations of the over 12,280 published scientific papers reporting on results of experiments with GeneTools custom products ordered by their customers over the decades. Over the course of more than 2 decades, GeneTools' crew of gene targeting experts and our crew of experts in producing precision-targeted antisense oligos have produced a wide variety of research tools. The following are of particular note. 1. A large family of practical research tools for use in the very demanding developmental biology field. Think zebrafish and a whole class of custom research tools not plagued by off-target problems. 2. One of our components was used successfully in the first experiment to treat progeria, a rapid aging disorder with a patient life expectancy of 15 years. 3. This component was also developed into the first ever FDA-approved treatment for Duchenne muscular dystrophy, now followed by 3 additional FDA approvals using this strategy to target other variants of Duchenne muscular dystrophy. 4. This component was successfully developed into a treatment for the childhood disease of spinal muscular atrophy, the major killer of children. 4. James Patrick Somerton, Organization of Information, focus on requirements still needed to complete the defeat of all human cancers. Substantial contribution in providing advice and assistance in drafting this letter. 5. Patricia Ann Somerton, patience and considerable assistance in making this presentation clean and compelling and keeping James's computer swearing to a minimum. Also serviced as an accountant for decades to support the development of new technologies through various companies. 6. Michael McAllister, validated LK1's elimination of cancerous cells through in vitro testing in 17 different human cancer cell lines. These cancer cell lines were composed of many different cancer types, including triple-negative breast cancer, ovarian cancer, choriocarcinoma, cervical cancer, lung cancer, colon cancer, gastric cancer, melanoma, glioblastoma, pancreatic cancer, and leukemia, which is a major cancer that devastates children. 7. Alan Schwartzman, business operations, public outreach, and assistance with organization of information. Where do we go from here? We are looking for your help in collaborating with us in pursuing further confirmation of the efficacy of the LK1 drug. We propose to provide quantities of the drug to interested collaborators and consultants to join us in validating the remarkable cancer-killing effects of the LK1 drug. In addition to seeking collaborations, we will pursue support from various institutions and foundations, including the Biden-Harris Cancer Moonshot Program, to provide advisement and logistical assistance in optimizing the drug for the use in humans. While pursuing this assistance, we are also working to gain approval to treat no-hope patients. These cancer patients are those who have been deemed terminal, no-hope patients, and are under hospice care. Since these patients have exhausted all other cancer therapies and treatments, treating no-hope patients will provide a clear opportunity to evaluate the LK1 drug's effectiveness and provide true hope to otherwise terminal patients. If any no-hope patients reading this paper would like to assist in the human testing of LK1, please reach out to the email address on the front page. We believe we have developed the capability to cure all human cancers. Due to the randomness of the mutagenic events that cause cancer, properties of these cancers may vary widely. While every cancer should be susceptible to our LK1 drug, because HCGB protein is the sole trigger for all human cancers, it will still need optimization of the treatment conditions to tackle the different cancer types. We ask that organizations dedicated to defeating cancer join us to bring this cure to no-hope patients. You may enjoy the following addendums that provides further information and support for our Cancer Cure program. Addendum 1, Morpholino, precise and efficient targeting tools. Morpholino antisense oligos. From 1985 through 1989, Dr. James Somerton pursued a project to radically redesign RNA, ribonucleic acids, with the explicit objective of incorporating special properties expected to be of value for pharmaceutical, therapeutic, and research applications. as well as avoiding properties expected to be detrimental for those applications. In 1989, that project culminated in a new class of products named morpholino antisense oligos, or just morpholinos. Each morpholino is designed to very specifically bind and block a complementary single-stranded RNA sequence under physiological conditions. The unique structure of such morpholinos is radically different from conventional nucleic acid-based antisense agents. Specifically, most conventional antisense oligos contain 5-membered ribose or deoxyribose backbone ring structures joined by negatively charged intersubunit linkages. In sharp contrast, morpholinos uniquely contain 6-membered morpholine backbone ring structures joined by neutral linkages to provide versatile utility. The unique structural properties of morpholinos, replacement of the ribose backbones with morpholine backbones, and replacement of negatively charged intersubunit linkages with non-ionic intersubunit linkages, provide a host of advantages over more conventional antisense oligos, including the following advantages. A. Resistant to enzymatic degradation, including within lysosomes. B. Does not generate an immune response. C. By far the greatest sequence specificity for their complementary RNA. Essentially free of off-target effects. E. Highly predictable targeting. F. Free passage between cytosol and nucleus of cells. G. Can alter splicing in the nucleus. H. Can block protein translation in the cytosol. I. Can block binding of regulatory proteins and non-coding RNAs. J. Stable at autoclave temperatures. K. Good aqueous solubility. L. Affordable due to cheap precursors, efficient assembly, and easy workup.